Hello again, grade 12 students. So in the previous video, we talked about the male and the female reproductive systems. These systems are responsible of producing mature sex cells, mature gametes, the sperm cell and the ovum. The sperm cells are produced in the testicles, in the male gonads, and the ovum is produced in the ovaries, the female gonads. When a sperm cell fertilizes an ovum, we get a zygote, which is the first cell of an embryo. Okay, the zygote will develop into an embryo. This first cell needs to divide many times to produce a multicellular organism. This division is called mitosis. So what is mitosis? Mitosis is a process by which a diploid cell divides many times to produce identical cells. So the daughter cells produced will be identical to each other and also identical to the mother cell. So we start mitosis with a cell that has 2N chromosomes, and we end mitosis with also a diploid cell having 2N chromosomes. Now, why do we need mitosis? As I told you, mitosis is important in the case of zygote development into an embryo. Another case is the growth of a tissue, an organ, as an individual, and also to renew and replace damaged body cells. Okay? Mitosis is one part of a cell's life cycle. So this part, the yellow part in here, is mitosis. Mitosis is one part of the whole life cycle of a cell. Another part is interphase. Interphase, let's say, we consider it as a phase to prepare the cell for mitosis. Interphase consists of 3 phases. We start with gap 1 or G1. In this phase, organelles of the cell are replicated, are doubled. During the S phase, it's the DNA that get doubled, that get replicated, synthesis phase. And G2, during G2, the cell makes sure that the DNA is replicated, and this phase is a preparation for mitosis. So interphase, 3 phases, one after another. We start with G1. During G1, the organelles are duplicated. This is synthesis phase, where the DNA is duplicated. Here, organelles duplicated, and here, DNA duplicated. And G2, this is a phase of preparation for mitosis. Now the cell is ready to undergo mitosis. Mitosis consists of 4 phases. The acronym PMAT refers to the 4 stages of mitosis: prophase, P for prophase, metaphase, anaphase, and telophase. During prophase, which is the first phase of mitosis, chromatin condenses. So the DNA coils, okay, coils more and harder around proteins to make visible chromosomes. The condensation of chromatin forms chromosomes, visible ones. We can clearly see their X shape, as you can see in here. Chromosomes are visible. Another thing is that the cell loses the membrane of the nucleus. So the membrane of the nucleus disappears, spindle fibers appear, and the centrosomes become asters. Okay, as you can see in here, a chromatin condenses. We can clearly see the chromosomes with their 2 chromatids. Okay, and that we lose the membrane of the nucleus. These are the asters. Centrosomes change into asters, and then the asters move to the 2 opposite poles of the cell. and we have the spindle fibers for sure. After prophase, metaphase occurs. During metaphase, chromosomes line up in the middle of the cell to form the equatorial plate, the metaphase plate in the middle of the cell. During anaphase, chromatids separate, as you can see in the picture. So here chromosomes are lined up in the middle of the cell. During anaphase, polar ascension occurs. Polar ascension, what is moving now? The chromatids of the chromosomes. Chromatids move to the opposite poles of the cell. After anaphase comes telophase. Telophase is the last phase of mitosis. In this phase, what happens is the opposite of what happened during prophase. During prophase, the nuclear membrane disappeared. Here, the nuclear membrane is reappearing, as you can see in the picture. Spindle fibers appear in prophase, but they disappear in telophase. In prophase, centrosomes became asters, but here asters are coming back to their centrosome state. During prophase, chromatin condenses to form chromosomes. Here, chromosomes do the opposite. They decondense back to the chromatin form. To end telophase, since now we have two groups of chromosomes, each one of one chromatid, okay, a cleavage furrow appears in the middle of the cell to split the cell into two daughter cells. So this is a cleavage furrow. The cleavage furrow splits the mother cell into two identical daughter cells. This process, when the cytoplasmic membrane, when the membrane of the cell constricts to divide the cell into two identical daughter cells, is called cytokinesis. So prophase, metaphase, anaphase, and telophase. Now we have two daughter cells with the same number of chromosomes, and mitosis is done. Keep in mind that during all phases of mitosis, we have the same number of chromosomes. During G1 of interphase, since we started with a diploid cell, so we have two chromosomes, but these chromosomes have one chromatid. During synthesis phase, we duplicate the quantity of the DNA, but the number of chromosomes does not change. So we have two chromosomes, but now these chromosomes have two chromatids. In G2, nothing happens. The cell makes sure that the DNA is replicated and ready. So we have two chromosomes, two chromatids. During prophase, chromosomes become visible, two chromosomes with two chromatids. We still did not divide the cell into two cells or separated the chromatids. Metaphase, the chromosomes are lined up in the middle of the cell, in the equatorial plate, so two chromosomes, two chromatids. Anaphase, chromatids separate, but we are still in the same cell. So both chromatids are present in the cell. Now, during telophase, we separated the two cells. So each group of chromatid is in the nucleus of one daughter cell, of each daughter cell. So we have two chromosomes, but with one chromatid. Okay, pay attention that during the synthesis phase of interphase that comes before mitosis, the DNA is replicated, but we didn't change the number of chromosomes. We duplicated the chromatids. In all the phases, the number of chromosomes is equal to 2n. So all cells in here are diploid. Okay, this is why we say, Aslan, look, in G1, we started with a cell with two chromosomes and one chromatid. And we ended with daughter cells with two chromosomes and one chromatid. The cells are identical to each other, and they are also identical to their mother cells. Now, mitosis happens in cells of animals and also cells of plants. There is little differences between mitosis of plants and mitosis in animals. This is because plants have no asters in their cells, so no asters in mitosis of plants, and also there is no cleavage furrow. We cannot split the membrane of the cytoplasm, the cytoplasmic membrane. Okay, look right here. This is an animal cell. A cleavage furrow constricts the cytoplasmic membrane to split the mother cell into two daughter cells. But look at the plant cell. We cannot split it. We cannot constrict the cytoplasmic membrane since plants have, or cells of plants have a cell wall, which is hard, which is rigid. So what happens to split the cell during cytokinesis is that a cell plate appears in the middle to split the mother cell into two daughter cells. Another type of division is meiosis. In both mitosis and meiosis, we start with a diploid cell. But now, the function of meiosis is to produce gametes, to make the sex cells, which are the sperm cells and the ova. This is the function of meiosis. So out of a diploid cell, we get haploid cells. Before meiosis, interphase happens, G1S and G2. During the synthesis phase, the DNA is replicated, so we will get two chromatids, and we have for sure G2. Meiosis consists of two big stages. We have meiosis 1, which is reductional. We are reducing the number of chromosomes. Instead of having two N chromosomes, the daughter cells will have N chromosomes. The second meiosis is equational. We mean that we are separating the chromatids. So it's the same number of chromosomes, but instead of having two chromosomes will have one chromatid. Let's start talking about phases of each meiosis 1 and meiosis 2. It's the same order. Meiosis 1, PMAT1, يعني prophase 1, metaphase 1, anaphase 1, telophase 1. During prophase 1, the nuclear membrane disappears, spindle fibers appear, centrosomes change into asters, and the chromatin condenses, so we can clearly see the chromosomes in their X shape. What happens is that in the cell undergoing meiosis, homologous chromosomes, they will pair up. They will stand next to each other, they will pair up, and this will allow for crossing over to happen. What do we mean by crossing over? Crossing over is the process of exchanging part of the DNA to increase variety in a population. Look at these two homologous chromosomes, one coming from the mother, one coming from the father. These homologous chromosomes, they exchange parts of their DNA just to increase variety. We call this process crossing over, and it happens in prophase 1 of meiosis 1. After prophase comes metaphase, the homologous chromosomes line up in the middle of the cell. During anaphase 1, pay attention in here, we have the homologous chromosomes separating. So each chromosome moves to a pole of the cell. For sure, we call this process polar ascension, but during anaphase 1, we are separating the homologous chromosomes, the chromosomes and not the chromatids. Okay, the last part, it's telophase. First, the membrane of the nucleus reappears, the asters return to the centrosome, form spindle fibers disappears. It's the opposite of what happened in prophase 1. Then we have a cleavage furrow that constricts the cytoplasmic membrane and splits the mother cell into two daughter cells. These daughter cells are now haploid. So the number of chromosomes is reduced. This is why we call it reductional division. The number is reduced. So we started with a diploid cell with 2 N chromosomes, and we ended with a haploid cell having N chromosomes. Okay, so the daughter cells are different than the mother cells. Daughter cells are not identical to the mother cell. No, they are different than the mother cell. After meiosis 1, meiosis 2 starts. So as I was saying, this is prophase 1. We have the 2 homologous chromosomes pairing up. Okay, look at them exchanging their DNA. So this is crossing over happening during prophase 1 of metaphase 1, of meiosis 1. At metaphase 1, the homologous chromosomes line up to form the equatorial plate. During anaphase, what separates the chromosomes? The homologous chromosomes separate to the opposite poles of the cell. So at the end, we have 2 daughter cells having half the number of chromosomes. We started with a diploid cell with 2 N chromosomes. We had 4 chromosomes at the beginning, and we ended with 2 haploid cells having half the number. This is meiosis 1. After meiosis 1 comes meiosis 2, the same order of stages. Prophase 2, أكيد, spindle fibers appear, the membrane of the nucleus disappears, centrosomes become asters, and we can for sure clearly see the chromosomes inside the nucleus. During metaphase, the chromosomes line up to form the metaphase plate. During anaphase now, the chromatids separate and moves to the poles of the cell by polar ascension. So at the end and during telophase, the cell will have chromatids, okay? Chromosomes of 1 chromatid. During telophase, asters return to the centrosome state, spindle fibers disappear, membrane of the nucleus appears, chromosomes decondense and go back to the chromatin form, and we have chromosomes of 1 chromatid at each side of the cell. وأكيد we have a cleavage furrow splitting the cell into 2 daughter cells. Let's look at the diagram showing meiosis 2. This is meiosis 2. Meiosis 1 gave us 2 daughter cells. Now, each daughter cell will continue meiosis 2. Here we have 2 chromosomes in the nucleus. The membrane of the nucleus disappears, spindle fibers appear, asters, the asters move to the poles. We have the chromosomes lined up in the middle of the cell, and then the chromatids separate. So at the end, as you can see, we have 1, 2, 3, and 4 haploid cells. These cells have N chromosomes with N chromatid, with 1 chromatid. So at the end of meiosis, we will get 4 gametes, 4 haploid cells. This is a table showing or comparing mitosis to meiosis. First, what's the function or what's the importance of going through mitosis? As I told you, mitosis for the zygote to develop into an embryo, for the body to grow, for the renewal, the replacement of damaged cells. While meiosis gives us the sex cells, the gametes. Mitosis happens in all the cells of the body except the sex cells, while meiosis is for the gametes, for the sex cells. Mitosis, it's one division. At the end, we will have 2 daughter cells, while meiosis, 2 divisions. And the meiosis 1, meiosis 2, and at the end, I will have 4 gametes. Here, 1 division, and before this 1 division, we have interphase. Here also 1 interphase, but before meiosis 1 and meiosis 2, there is no interphase between meiosis 1 and meiosis 2. No crossing over. There is crossing over where the homologous chromosomes exchange their DNA. Mitosis gives us 2 daughter cells that are identical to the mother cell. And as I told you, same number of chromosomes in all phases of mitosis. Okay? Here, the number of chromosomes is reduced into half after meiosis 1. And then after meiosis 2, we separate the chromatids. So we start with a mother cell, 2N, diploid mother cell, and we end with a haploid gamete. This is all about meiosis and mitosis. Meiosis and mitosis have an important role in the formation of the sex cells. So you will see that mitosis and meiosis happen in spermatogenesis, which is the process of formation of sperm cells. وكمان بيصيروا بالoogenesis, which is the process of formation of ovum. Okay, so I'll see you in the upcoming